welcome to the deep dive. Today we're jumping into a foundational text, Porth's Pathophysiology, and focusing on Chapter 51. We're looking at the body's largest organ, the skin, or the integumentum.
Yeah, it's huge. It makes up something like, what, 16% of your total body weight? It's this massive interface with the world outside.
Exactly. And our goal here is really for you, the learner listening in, to get a solid grip quickly on the key structures, the cells involved, and, the crucial functions of the skin described in Porth.
We want to make this dense chapter feel much more digestible, and it's fascinating because the skin isn't just a barrier. It's often the first place we see signs of trouble brewing inside.
A diagnostic window, you could say.
Absolutely. That's a central theme. Think about systemic diseases, things happening deep within the body. They often show up on the skin first.
Like the classic melarash in lupus.
Precisely. Or that sort of bronze tint you see with Addison disease or jaundice signaling liver problems. Understanding the normal skin structure is step one, because pathology is really just that normal structure going wrong.
Okay, so let's dive in. We'll look at the layers, meet the specialized cells, kind of like the skin's internal team, and then unpack its vital jobs, from sensing touch to controlling temperature.
Sounds good. So structurally, you've got three main layers to think about. There's the outer epidermis, then the dermis beneath it, and finally the subcutaneous fat layer anchoring everything.
And between the epidermis and dermis, there's that really important dividing line, the basement membrane.
Crucial, yes. Let's tackle the epidermis first. It's the outer shield. Importantly, it's avascular, no blood vessels of its own.
So it gets all its nutrients by diffusion from the layer below.
That's right, from the dermis. And it's mostly built from these cells called keratinocytes. They make up maybe 85% of it. They're stratified, squamous epithelial cells.
And these keratinocytes, they're not static, are they? They're on a journey.
A constant journey, yeah. It takes about 20 to 30 days for one to be born deep down and make its way to the surface. This journey creates the different layers or strata of the epidermis.
Okay, walk us through those strata, from deep to superficial.
Right. It all starts in the stratum basale, sometimes called the germinativum. This layer sits right on the basal lamina. It's where the cell division mitosis happens constantly.
The keratinocyte factory.
You got it. And because of that high division rate, it's also where you often see basal cell carcinomas start.
Makes sense. So they divide there, then what?
Then they start moving up and changing. The next layer is the stratum spinosum. They call it the prickle cell layer because the cells start developing these connections, these desmosomes, that look kind of spiny under a microscope.
Okay.
Above that, the stratum granulosum. This is a really key layer functionally. The cells here are starting to die, but before they do two crucial things. They synthesize a lot of keratin, the protein that makes skin tough, and they produce and secrete these things called lamellar bodies.
Lamellar bodies. Why are those so important for us to remember?
Because they contain lipids, fats. They release these lipids into the spaces between the cells, creating the skin's essential waterproof barrier.
Oh, so that's the seal that prevents dehydration.
Exactly. And it's why in something like a major burn where you lose this layer over a large area, fluid loss becomes a life-threatening problem. That barrier is gone.
Got it. So granulosum is key for waterproofing. What's next?
Then in thick skin, like your palms and soles, there's a thin, clear layer called the stratum lucidum. It's not everywhere.
Okay.
And finally, you reach the top, the stratum corneum. This is the layer you actually touch. It's composed of dead, flattened, keratin-filled cells. They're constantly shedding. This is your main physical barrier against microbes and water loss.
So that whole 20 to 30-day cycle, the keratinization process, needs to be perfectly balanced by production matching shedding.
Absolutely critical. If that balance is off, you get problems. Like, take psoriasis. In psoriasis, that whole cycle speeds up dramatically, maybe down to just a few days instead of weeks.
Leading to that buildup of skin cells, the plaques.
Precisely. The shedding can't keep up with the production.
Okay, so that's the keratinocyte journey. But there are other important cells scattered in the epidermis, too, especially down in that basal layer. The support team, you call them.
Yeah, the specialists. Let's start with the melanocytes. These are the pigment factories. They produce melanin.
The stuff that gives skin its color and protects from the sun?
Right. And melanin actually comes in two main forms. Eumelanin, which is brownish-black and offers the best UV protection, and pheomelanin, which is more reddish-yellow.
Now, here's something interesting from Porth's. It says that people with darker skin and lighter skin have roughly the same number of melanocytes. How does that work if the UV protection is so different?
Yeah, that's a fantastic point and often misunderstood. The difference isn't the number of factories, it's their productivity and packaging.
Okay.
In darker skin, the melanocytes produce more melanin, especially eumelanin, and they package it into larger granules called melanosomes. These larger melanosomes are then transferred individually to the surrounding keratinocytes and sort of form a protective cap over the nucleus.
Ah, shielding the DNA.
Exactly. In lighter skin, the melanocytes are less active, produce less eumelanin, and package the melanin into smaller melanosomes that often get bundled together. They just don't provide the same level of consistent cover. So, same number of cells, but very different functional output.
That's a great clarification. Structure impacts function right there. Who's next on the specialist team?
Next up are the Langerhans cells. These are the immune surveillance cells of the epidermis, maybe 3 to 5% of the total cells.
The security guards, maybe.
Or perhaps the intelligence agents. They have these long arms, dendrites, that reach out between the keratinocytes, sampling the environment.
What are they looking for?
Foreign invaders, antigens. When they find something suspicious, they grab it, process it, and then they actually leave the epidermis.
Leave. Where do they go?
They migrate to the nearest lymph node, and there they transform into potent antigen-presenting cells. They basically show the antigen to the T cells, kicking off a specific immune response.
So they're crucial for things like allergic reactions on the skin, contact dermatitis.
Absolutely. They initiate that kind of immune response, and they have a distinctive feature inside them, these Burbeck granules that look a bit like tennis rackets under an electron microscope.
Interesting detail. Okay, who else is down there?
We also have Merkel cells. These are less numerous. They're connected to nerve endings and function as touch receptors, specifically for light touch and texture.
So part of the sensory system. Where would you find lots of them?
In sensitive areas like fingertips, lips, and even around hair follicles, they form what's called the Merkel disc with the nerve fiber. There's also some thought they might have neuroendocrine functions, releasing hormones locally, but their primary role is sensory.
Okay. And before we leave the epidermis, how do all these cells stick together so tightly to form that barrier?
Ah, good question. Cell junctions are key. You have desmosomes, which act like spot welds, anchoring cells to each other via intermediate filaments called tunofilaments. These provide mechanical strength, resisting pulling and stretching.
And you also mentioned waterproofing.
Right, that involves tight junctions, which form seals between cells, preventing stuff from just leaking through the gaps. They really enforce that barrier integrity alongside the lipids from the lamellar bodies.
Makes sense. Okay, let's move deeper. We cross that basement membrane zone now. What's its main job?
Think of it as sophisticated double-sided tape. It anchors the epidermis firmly to the dermis below, but it's more than just glue. It's also a selective filter, regulating what passes between the two layers.
And clinically, this layer is a big deal, right?
Huge deal. Many blistering diseases, the bullous diseases, involve problems right here at the basement membrane.
How so?
While it has a complex structure layers like the lamina lucida and lamina densa, the epidermal cells attach to it via specialized structures called hemidesmosomes. If your immune system mistakenly attacks components of the basement membrane or these hemidesmosomes,
Layers separate.
exactly. The epidermis lifts off the dermis and fluid fills the space, creating a blister or a bulle if it's large. So its integrity is absolutely vital.
Got it. So if the basement membrane is the anchor, what's the dermis itself like?
The dermis is the support structure. It's a much thicker layer of connective tissue. It provides the skin's tensile strength, its resistance to tearing and elasticity, and crucially, it contains the blood vessels that nourish the epidermis.
And it has layers too, doesn't it?
It does. Two main ones. The upper thinner layer right next to the basement membrane is the papillary dermis. It has these finger-like projections, the papillae, that push up into the epidermis.
Like interlocking fingers.
Sort of, yeah. And these papillae are packed with capillary loops. That's where the diffusion of nutrients and oxygen to the epidermis happens.
Okay, and below that?
Below that is the much thicker reticular dermis. This forms the bulk of the dermis. It's characterized by dense, interwoven bundles of collagen fibers, which provide strength, and elastic fibers, which allow the skin to stretch and recoil.
And the way those collagen bundles are arranged, that matters, right? Like for surgeons.
Definitely. The collagen fibers tend to run in specific directions in different body areas, creating lines of tension. Surgeons try to make incisions parallel to these lines, Langer's lines, to minimize scarring because the wound edges come together more easily. Structure influencing practice.
Fascinating. Now, the dermis isn't just structure and blood supply. It's also where a lot of the sensory action happens.
Oh, absolutely. It's richly innervated. We talked about Merkel cells partly in the epidermis, but the dermis houses several key encapsulated nerve endings, specialized receptors.
Let's talk about those. Which ones do we need to know?
Okay, think about different types of touch and pressure. You have Pacinian corpuscles. These are large, onion-shaped structures found deep in the dermis and even subcutaneous tissue.
What do they sense?
They're sensitive to deep pressure and, importantly, vibration. They adapt very quickly, meaning they fire when the pressure starts or changes, but not if it's constant.
Like feeling your phone vibrate in your pocket.
Perfect example. Then you have Meissner corpuscles. These are found up in the dermal papillae, especially in sensitive, hairless skin like fingertips and lips.
Other job.
They detect light touch and texture changes. Like Pacinian corpuscles, they're also rapidly adapting. They help you feel the shape and texture of objects you're holding.
Okay, deep pressure vibration, Pacinian, light touch texture, Meissner.
What else? There are also Ruffini corpuscles, or endings. These are found deeper in the dermis and respond to sustained pressure, stretch, and possibly warmth, although their role in temperature is less clear than cold. Importantly, they are slowly adapting.
Meaning they keep firing as long as the stimulus is there.
Exactly. They tell your brain about ongoing pressure or skin stretch, like when you're grasping something firmly or your joints are moving. There are also free nerve endings that sense pain, itch, and temperature changes.
So a whole network of sensors. What about temperature regulation? The dermis is key there too, right?
Absolutely central. It uses blood flow primarily. Your sympathetic nervous system controls specialized connections between small arteries and veins in the dermis called arteriovenous anastomoses.
Shunts, basically.
Exactly. When you need to lose heat, these shunts close, forcing blood into the superficial capillary loops near the skin surface where heat can radiate away.
And when you're cold.
The shunts open wide, allowing blood to bypass those superficial loops and stay deeper, conserving heat in the body's core. It's a very efficient system. Plus, you have evaporative cooling from sweat glands, which we'll get to.
Right. And before we move to appendages, what about the dermis's immune role? We mentioned Langerhans cells migrating from the epidermis.
Yes, and the dermis itself has its own resident immune cells. You find macrophages, which clean up debris and pathogens. You find T lymphocytes, including memory T cells, which are important for remembering past infections or allergens, playing a role in delayed type hypersensitivity.
Like the reaction to poison ivy.
That kind of thing, yes. And very importantly, you have mast cells. These are strategically positioned near blood vessels.
What's their big role?
They are packed with granules containing histamine and other inflammatory mediators. They degranulate rapidly in response to allergens or injury, causing the redness, swelling, and itching of immediate hypersensitivity reactions, like hives, or an allergic reaction to a bee sting. You also find dermal dendrocytes, which are another type of antigen-presenting cell.
So layers of immune defense throughout. Okay, let's talk appendages. These are structures anchored in the dermis or subcutaneous layer, but often extend outwards. Sweat glands first.
Sure. Two main types. The most numerous are the eccrine sweat glands. They're found almost everywhere on the body.
And their job is mainly cooling.
Primarily, yes. They open directly onto the skin surface via a pore, and they secrete a watery sweat made mostly of water and salt. Evaporation of this sweat cools the body.
Okay, and the other type.
The apocrine sweat glands. These are larger, and they're mostly found in specific areas like the armpits, groin, and around the nipples. Unlike acrine glands, they typically open into a hair follicle, not directly onto the skin surface.
And their sweat is different.
It is. It's a thicker, milky secretion that contains lipids and proteins. It's actually odorless when first secreted.
So where does body odor come from?
From bacteria living on the skin surface. They metabolize the components of apocrine sweat, producing the volatile compounds we perceive as body odor. These glands become active at puberty, linked to hormonal changes.
Speaking of hormones, that brings us to sebaceous glands.
Right. These glands are almost always associated with a hair follicle, forming the pilosebaceous unit. They produce an oily substance called sebum.
What's sebum for?
It lubricates the hair and the skin surface, helping to keep it supple and providing some waterproofing.
And the key link for pathophysiology here is
Hormones again, specifically androgens, like testosterone. Androgens stimulate the growth and secretory activity of sebaceous glands.
which is why acne often flares up during adolescence or other times of hormonal change.
Exactly. Acne vulgaris involves inflammation and blockage of these pilosebaceous units, often driven by increased sebum production linked to androgens, plus bacterial involvement and abnormal keratinization within the follicle.
Makes sense. Okay, let's quickly cover hair and nails.
Sure. Hair is essentially a filament of dead keratinized cells produced by the hair follicle. It grows in cycles. Anagen is the active growth phase, catagen is a brief transitional shrinking phase, and telogen is the resting phase before the hair eventually sheds.
And a little muscle attached.
The arrector pili muscle. It's smooth muscle controlled by the sympathetic nervous system. When it contracts due to cold or fear, it pulls the hair follicle upright, causing goosebumps. It's thought to be a leftover thermoregulatory mechanism trapping air.
And nails.
Nails are tightly packed plates of hardened keratinized cells. Unlike hair, they grow continuously from the nail matrix, which is the area under the proximal nail fold.
And they're useful diagnostically.
Very much so. You can assess capillary refill for circulation, look for clubbing, which might indicate lung disease, check for splinter hemorrhages, or see pitting associated with psoriasis. They're like little windows onto systemic health.
Okay, we've covered a lot of ground layers, cells, appendages. Let's pull it all together. What are the absolute key functions of the skin we need to take away from this deep dive?
Well, number one is clearly protection. It's a physical barrier, a chemical barrier with that slightly acidic surface, and an immunological barrier.
Right, and temperature regulation through blood flow control and sweating.
Definitely. And sensation, touch, pressure, vibration, pain, temperature, it's our main interface for interacting physically with the world.
What about functions we might not immediately think of?
Okay, a really big one is its endocrine function, specifically vitamin D synthesis.
How does that work again?
There's a precursor molecule in epidermal cells, 7-dehydrocholesterol. When UV light from the sun hits the skin, it converts this molecule into cholecalciferol, which is vitamin D3. This then travels to the liver and kidneys to be activated into the hormone calcitriol, which is essential for calcium absorption.
So the skin kicks off a vital endocrine pathway. Anything else?
Minor roles in excretion, you lose some salts, urea, even CO2 in sweat. And absorption, while it's mainly a barrier, lipid-soluble substances can be absorbed through the skin.
Which is how transdermal patches for medication work.
Exactly. But it also means toxins can potentially be absorbed too.
So to wrap up for the learner, key takeaways are the dynamic journey of keratinocytes creating that vital barrier, the specialized security team like Langerhans cells and melanocytes, and the dermis as this complex hub for structure, sensation, and temperature control.
Absolutely. And recognizing how tightly integrated it all is, the structure directly enables the function, and disruptions in structure lead directly to pathology.
It really drives home that the skin isn't just a covering. It's a complex, active organ system.
It truly is. And maybe a final thought to connecting back to that neuroimmune link we touched on. We know stress, acting through nerves, can influence skin immunity, maybe worsening acne or eczema.
Right.
So the question becomes, what other internal states, stress, inflammation, metabolic changes, are constantly communicating with your skin, potentially altering its structure or function in subtle ways, even before a visible sign appears? The skin is always reflecting what's happening inside.
A really powerful idea. The skin as a continuous readout of our internal well-being. Fantastic. Thank you for walking us through that.
My pleasure. It's a fascinating system.
And thank you all for joining us on this deep dive into the skin's structure and function based on Porth's. We'll catch you on the next one.